biotech and nutritional companies, and in 1997 he joined Dr. Stock at California's UCLA's School of Medicine and Technology Society with the specific aim to study aging and the possible interventions to delay the onset of degenerative diseases. Since 2000, he has held the position of Vice President of Research and Development at the Kronos Group. Informing us today about the importance of biomarkers and laboratory testing in anti-aging medicine, please welcome Dr. Christopher Heward.
Thank you, Phil. Well, first of all, I'd like to thank the conference organizers for inviting me to come and speak to you all today on my favorite subject, which is the science of trying to help people live healthier, longer lives. Before I start, I'd like to just get a show of hands. How many physicians are in the audience? Okay. How many are offended by off-color jokes? Oh. Well, you can come and hit me later. I'm going to tell it anyway. I like to tell a joke at the beginning of a talk to kind of mostly relax myself. And my favorite joke in these kinds of talks is about this old guy who comes to the doctor's office. And he's kind of nervous, and the doctor asks him, well, what can I do for you, sir? And he says, you know, doc, I've got this problem. I can't pee. And the doc says, tell me about it. And how long has this been a problem? He says, well, it's been growing for a number of months. At first it started where the flow was less and less, and I had a harder time going, and it was kind of painful. And then yesterday I just couldn't pee at all. And so you got to do something. You have to help me, doc. And the patient and the doc says, well, you mind if I ask how old you are? And the guy kind of stood up straight and very proud. He says, doc, I'm 101 years old. And the doctor said, well, then you've peed enough. The reason I like that joke is it illustrates two problems that we in this field of anti-aging medicine have to face. One is that people tend to wait until they have a serious problem, a health problem, before they come to see the doctor, rather than coming early in trying to take a proactive approach to their health. And that's a big mistake, and part of our burden as professionals in this field is to help people understand that they need to come in early in the game and start taking care of these progressive degenerative problems that are going to be serious later on if they don't. The other issue is a philosophical one. I mean, there are actually people out there who believe that we shouldn't be doing anything to because you're supposed to grow old and die. And this joke kind of illustrates how ridiculous that is. I mean, who's to say when you've peed enough? Certainly not me. Okay, enough of that. I should tell you a little bit about my background. I am a research endocrinologist. I'm a biochemist, and so my bias, being a research scientist, will show up in some of the presentation. To start off, I think I should tell you a little bit about the company I work for, which is the Kronos Group. And it's basically a group of scientists and physicians and business people who have gotten together to try to figure out how to help people live healthier longer. And as you all know, that's not a very easy job. There's not as much science as we would like there to be in this area, and we're trying very hard to push out the frontiers of our knowledge so that we can do a better job of helping people do just that as time goes by. What we're really talking about here is trying to extend this curve. This is a curve that depicts the average life expectancy at birth by year. And as you can see, we've been doing a pretty decent job in the last century. The average life expectancy has increased from about 47 years to about 76 years, with females doing consistently better than males during that period. That's a pretty impressive change in 1 century, considering how long it took to get from 20 years to 47 years prior to that. The problem is in extending this curve, and you can see by looking at the trend line that although the slope at the first part of the century was pretty good, towards the latter part of the century it's kind of leveling off. And there's some debate among epidemiologists and gerontologists about whether it's even possible for us to continue to extend that curve upward. And certainly I think the most widely held view by gerontologists is that if we're going to actually improve or continue to move that line up, we're going to have to actually begin to impact the aging process itself. It's not going to be good enough to just impact diseases that kill us, but in fact we have to get at the fundamental root causes of aging. So that's kind of my challenge as a gerontologist, to try to get in and figure out how we can actually do that. Unfortunately, before we figure out how we can combat aging, we have to answer this question: what is aging? And the sad reality is that we don't know. Yeah, I certainly don't know. I've been studying aging for 15 years, and I'm increasingly discouraged by how much I know about exactly what's going on in a fundamental way. Still, having said that, there are some encouraging things. And this particular slide I like to show because this slide is one that I read some 15 years ago, and it was one of the first things that got me interested in the aging process itself. The thing about it is this is a list of mammals mostly, and it shows their maximum lifespans. And the thing that struck me about this slide is that all mammals seem to age in roughly the same way. You can see dogs don't make it much past 20, and men don't make it much past 120. But we age differently. If I told you I had an 18-year-old dog, what would you know about that dog? He's a rickety old guy, isn't he? He's on his last legs. If I told you I had an 18-year-old son, born the same day as the dog, what would you know about that boy? Prime of life, hitting his stride. Same 18 years, different biochemistry. Well, that means aging is biochemical. That's exciting, because if it's biochemistry we're talking about, and we can understand that biochemistry, maybe we can get control of it and regulate it. That's kind of exciting. Another thing that struck me about this slide is the tortoise. That is a big animal, or can be quite big. Suppose I told you I had a 120-year-old tortoise. What would you know about that tortoise? Is it a rickety old tortoise? No. Tortoises manage to live to be very, very old and don't seem to show any of the same sorts of degenerative processes that we commonly recognize as aging in humans. So it's an indication that there is a biochemical solution, at least for the tortoise, that avoids some of these problems. And again, maybe we can learn about that, and as we learn about it, can apply some of that technology in our own lives. So that's really what encouraged me after many years as an endocrinologist to get into gerontology and begin to study aging itself. So here's my model of, this is kind of a working hypothesis about aging. This is how I think it works. We start out with oxidative metabolism. In order to get the energy we need to survive, we have to oxidize food, and in the process of doing that, we generate free radicals. Those free radicals are highly reactive molecules that can do damage to vital biomolecules in the body. Primarily, the most important ones as far as aging is concerned are probably nuclear and mitochondrial. When damage is done to these biomolecules, the cells in which these molecules function are impaired. And as the functional capacity of the cells declines, they exhibit this characteristic called disdifferentiation, where they no longer behave the way they were originally intended to behave and begin to disdifferentiate, expressing characteristics that are less like the fully differentiated cells that they were genetically supposed to be. As the cells decline in their function, the tissues in which those cells reside also decline, and the organs that they reside in also begin to lose their proper function. How many people in here are as hot as I am? This is really warm. Do you mind if I take my jacket off? as the, sorry, I'm trapped like a rat. Okay, I think I messed up my, oh, here we go. How's that? Can you hear me? I messed it up. Sorry about that. Yeah, good idea. If we're gonna, sorry about that. Oh, there it is. Wait. Thank you. Okay, that's better, a little better. So anyway, as the organ systems declined, the systems, or the organs, as the organs decline, the systems in which the organs reside begin to decline in function as well. And one of the most important systems that is part of that declining process is the neuroendocrine system. And since the endocrine system is responsible for maintaining the constancy of the internal environment and homeostasis, as the endocrine system declines, we get impaired homeostasis, and the hormonal balance shifts to an imbalance, and the stability of the internal environment diminishes. And when the internal environment in which the cells are attempting to survive changes to an environment which is not conducive to cellular health, then cell function declines further, and you have this vicious cycle that just spirals out of control until you grow old and die. Unfortunately, there's not a whole lot we can do about intervening in these processes except try to struggle to maintain hormonal balance, and that's one of the primary targets that we use in addressing the problems of aging once they begin to occur. As far as prevention is concerned, we can try to intervene early in the game and try to prevent some of the oxidative stress, or intervene later in the game and try to repair or induce some of the repair mechanisms that will correct the problems, the damage that's occurred. So that's kind of aging in a nutshell. As you can see, it's kind of a hard problem. There's not a lot of handles to grab a hold of for intervening in a targeted way to solve the problem. None of that is very important to the patients that present to anti-aging physicians in terms of what they care about when it comes to aging. What they care about are these things. They've got all of these symptoms of aging which are plaguing their lives, and they would like to see these things corrected, and they'd like to see them corrected now. If the symptoms of aging, if the aging process is allowed to continue unchecked, those symptoms escalate to the common progressive degenerative diseases associated with aging, and this is a partial list, and these are the things that if we're not successful in checking their progression, they will ultimately end up as the ultimate causes of our death. 3 out of 4 people in this room will die of age-related diseases. And at this point, there's not a whole lot we can do about that except intervene in a targeted way and try to slow the process down. What that means is that anti-aging medicine, in terms of true interventions that combat the aging process in any fundamental way, doesn't exist yet. We don't know how to do it. So we're stuck with some pale approximation of anti-aging medicine, true anti-aging medicine, which is really high technology preventive medicine. So what's my vision? My vision is that eventually we will be able to come up with interventions that actually will enable us to intervene and slow down the aging process itself. In the meantime, what can we do to help ourselves, given the technology we have available? The best thing we can hope for is to try to extend our healthy lifespan so that we can buy some time, hopefully enough time that will enable us to live to a period where some true anti-aging technologies are available. Looking at the data from the Baltimore Longitudinal Study on Aging, it gives us some clues about how we can go about extending our own personal lifespans given current technology. The way the BLSA study is structured is they take individuals and they test them using a broad spectrum of biochemical and physiological parameters and monitor them longitudinally to see how people age over time. And this is the general pattern that you see. This is just 4 of the parameters that are looked at. They look at a great many parameters, and they all follow pretty much the same pattern. And so if you want to generalize from the specifics to a general pattern, you get kind of this shape. And the point I want to make about this pattern from the Baltimore study is look at this incredible plateau of optimal health here, of maximum functional capacity. There isn't one. We peak and we start immediately back down, and it's pretty relentless from that point forward as a function of age. The interesting thing is, if you look at the average data of the average person in the Baltimore Longitudinal Study, it looks as though we ought to be able to anticipate a life expectancy of somewhere in the range of 100 years. But we know from one of the earlier slides that life expectancy is nowhere near that. Why not? Well, the answer is due to the fact that not only do we not all age the same as individuals, but even within our own bodies, we don't age uniformly, so that some parts of our bodies age faster than others. And here's an example of an individual whose cardiovascular system is declining in its functional capacity prematurely. So rather than following this white path down towards a lifespan of about 100, his cardiovascular function is declining at a rate that's going to bring him below this threshold of viability prematurely. And this person's going to die at about age 60, 65 and be cheated out of 30, 35 years of the lifespan that he is genetically endowed with, simply because he's not aging uniformly. If you look at centenarians to find out how these successful agers manage to do it, and they do autopsies on them to find out what's wrong with them, it's not that they didn't age like the rest of us, they just managed to age uniformly. So when you look in there and find out what's wrong with them, everything's wrong with them, but just nothing in particular. In this case, this individual has something in particular wrong with them. And traditional medicine, the conventional approach, is to wait until the functional capacity has dropped to a point where it reaches full disease status, and then intervene in a targeted way to try to buy a few extra years. The problem with that approach is you buy a few extra years at this very low level of functional capacity, which is, although you're increasing, if you're successful, the quantity of life, you may not be increasing the quality of life much. A better approach, I think, is to intervene early. Identify as quickly as possible these opportunities where you can detect a premature decline in functional capacity of one or another system. And cardiovascular disease is an important one because it's the #1 killer in the United States. And if you intervene early enough and can correct the problems and reduce the risk factors, you may be able to restore a more youthful pattern of decline and enable the individual to live out, again, something closer to the full complement of years that they're genetically able to live. And that's, in principle, the approach that we take in trying to apply our optimal health program at Kronos. And fundamentally, because I'm kind of a rigorous, stodgy, conservative, scientific type, we're science-based. Nothing we do at Kronos is based on anything but solid scientific foundation. That limits us a little bit in terms of what we do, but I think it's the safest way to practice this type of medicine if our goal is to really extend the healthy lifespans of our patients. Our paradigm is really assess and intervene rather than diagnose and treat. We're not looking for diseases, we're looking for disease processes, and we're trying to catch them as early as possible and intervene in as targeted a way as possible to head off problems in the future. And our focus is on prevention. We do that by detecting diseases as early as possible. We also focus as best we can, and I'll talk a little more in detail about oxidative stress, simply because we believe that that's a key component of the fundamental mechanisms of aging. Unfortunately, as you'll see, we don't know that much about it. We do, since I'm an endocrinologist, feel pretty comfortable in getting aggressive about maintaining youthful hormonal balance. We also have a nutritional program, which is pretty straightforward, and no optimal health program is complete without an exercise component. And I'll go through each of these in some detail one at a time. The assessment of disease risk, it really involves a wide variety of biomarkers that we measure in our laboratory. We look at over 150 biochemical parameters, and based on that, we get a pretty good indication of what the health risk status is of the patient in terms of a wide variety of potential health problems down the road. It's amazing how frequently we find problems even in relatively young people. In fact, I don't think there's an example of a patient who's been through the Kronos program where we have not found a problem. Even in 28-year-olds, there's an indication that there's some kind of degenerative process going on that demands intervention now to prevent a problem down the road. So we don't just wake up one day and have osteoporosis and start fracturing. That osteoporosis started when we were 30. If we can identify it when we're 30 and head it off at that point, it won't be a problem ever in our lives, because there are very simple, straightforward interventions that can prevent it from becoming fracturing osteoporosis later on. And there are many more examples of that. And the biomarkers enable us to do a good job of monitoring that process. With 150 parameters to look at, I can't go into much detail about that. I would like to comment briefly on the cancer risk assessment, which we don't do a very good job of, but are currently doing research in our research laboratory to develop tests that will do a very good job of that. And I think probably next year, if I speak at this conference, I will talk about that test, because it's showing some very promising results. In addition to that, genetic testing is something that one of our sister companies is looking at. We don't do genetic testing yet, simply because I think it's difficult to do it well given current technology, and in a preventive health environment, there may be an application for it as soon as we can work out the ethical and counseling issues that have to be addressed in order to do a good job of that. The technology is coming fast, and as we get more and more information, we'll be better and better at applying that new technology. This is just an example of the Kronos laboratory report. It's kind of a unique report, because in addition to the regular reference range values that are shown over to the right, we have these green and red bars that give the patient a visual indication of what the information means, and it kind of helps the patient become more involved in what their personal results are, and helps them better understand what the numbers are, and they get committed to improving those things that seem to be out of range and bad. You find Lp(a) here as an example of a number that's in the red, and even the patients can tell that's a bad thing, whereas the triglycerides are down low enough in the green, so they don't worry about those so much. We also, in addition to the lab tests, look at a broad spectrum of physiological parameters. We do a treadmill test and the EKG. We measure basal metabolic rate. We do strength and flexibility testing. We measure body composition using DEXA. We do use the H-scan. We also do glaucoma screening, hearing testing, and cognitive function testing. I should mention that cognitive function testing is the weakest of these links. It's very difficult to measure cognitive function in a way that will get you information that is able to pick up the subtle declines in function that occur with aging, say between the ages of 50 and 65, and yet is not so sensitive that it gets confused by a bad day. The other part of that is that you need to have an automated test that's very consistent and one that you can deliver consistently in about 15 minutes. Otherwise, it's cost prohibitive to do repeatedly on your patient population. So we're working with the Department of Defense that's developed a test at West Point for measuring concussion in cadets who take boxing. And we're trying to apply it in our application. We're doing basic research on that. And as the data are collected, it's looking increasingly promising, but we're not quite ready to put that out to the world yet. So that's coming. We also do health screening by CT, and we do all of these types of scan, the heart scan, the lung scan, the whole body scan, and virtual colonoscopy. But I have to say that for my money, although there are cases where it's appropriate to do all of these, the only one that I really feel comfortable using is the heart scan, because when an individual presents with a high-risk blood lipid profile, you need to be able to tell whether there's frank disease, and the CT scan is a very good way of doing that. The problem with the CT is that it uses ionizing radiation, and it uses pretty hefty doses, and we all know that ionizing radiation causes free radicals, and free radicals are not good for you. So we have to use this technology carefully, and in only those cases where we think that the risk-benefit analysis falls down in favor of the patient's benefit. Reduction of oxidative stress. Ox stress is kind of near and dear to my heart. We've been studying it very hard for the last few years, trying to develop a paradigm like this. We still haven't been successful. It's been very hard. We have some of the best scientists in the world in our group that are looking at ox stress, and we have lots of heated arguments about the data that we generate. The idea, though, is to try to identify ways of measuring oxidative stress so you can tell people who are experiencing above average damage and have below average protection factors, say somebody like this person. And the idea would be to develop interventions that will enable you to go in and change their status to a person with below average damage, and presumably the way you would do that is by increasing their protection. We look at a wide variety of protection factors, most of which are antioxidants, either internal or external. And at the same time, we also look at a number of damage factors, or pro-oxidative damage factors, including serum iron. We have a much longer list than this, but these are the good ones. Lipid peroxidation, the total alkanals and isoprostanes, protein oxidation products, carbonyls and dityrosine. We do these last three on the LC mass spec. It's a very, very good assay, good way of getting hard data. Once we've figured out that these measurements actually are of value, we will develop ELISA assays so they're made available out to the world, and then we can use much less expensive testing to answer some of these questions. Just for the sake of discussion, I want to concentrate on these last two DNA damage products. And the reason for that is that we've just completed some testing on that, trying to validate that, in fact, measuring these things tells us something about the oxidative stress level in our patients. The idea is this: if we come up with these measurements of oxidative stress, but these measurements can't tell the difference between populations that we know are under high levels of stress and populations that aren't, then they're not of much value to us in a clinical setting dealing with healthy patients. So we know, for example, that smokers are under a very high level of oxidative stress, and these measurements ought to be able to tell the difference between a person who's smoking and a person who isn't. So we did a little study, and the study had these assumptions. Smoking increases DNA damage markers. 8-OHDG is the best indicator of DNA damage, and that we just pulled out of the literature. The oxidative stress community believes 8-OHDG is it's probably the most studied DNA damage indicator. All types of DNA damage are repaired quickly and efficiently. Repair is almost perfect. That's an assumption that's repeated in the literature over and over again. And therefore, an increase in urinary damage levels is indicative of increased damage and not increased repair. The reason I emphasize this point is that if we're measuring these products in the urine, they got into the urine because the body recognized the damage, excised it, cleared it into the blood, the kidney then cleared it into the urine. So we're actually measuring it after it's being repaired. So is an increase in DNA damage products in the urine an indication of increased damage or increased repair? The answer is, if repair is very efficient, almost 100%, then any increase in the urine is an indication of increase in damage, not increase in repair. And that's the assumption that exists out there in the literature. So we just proceeded on that assumption and did our little study. And we had 50 males and 38 females about age 50. We had 45 never smokers, 14 ex-smokers, and 29 smokers. The ex-smokers had, on average, not smoked for 15 years. And here's the data we got, which surprised us very much. Notice with 8-OHDG that there's a statistically significant difference between the non-smokers and both the ex-smokers and the smokers, but no difference between ex-smokers and smokers. What that means is that the people who used to smoke 15 years ago are still showing elevated levels of 8-OHDG in their urines. And the only way that can possibly be true is if they're still repairing 15-year-old damage today. That's an amazing result. If you look at 5-hydroxymethyluridine, which is another type of DNA damage adduct, we don't see that same pattern. The difference between the ex-smokers and the non-smokers is not significant. Only the current smokers show higher levels of 5-hydroxymethyluridine. So that's kind of an exciting result because it gives us an opportunity with one test or two tests, looking at two adducts, to gauge whether DNA damage has occurred recently or a long time ago when we find it elevated. Now, obviously, there's a lot more work to do. We've got more validation studies in process. We're going to look at ionizing radiation. We're going to follow airline pilots and flight attendants. We're going to look at people who are exposed to medical radiation. We're going to look at people who are hyperthyroid. There's a lot of other ways of validating these tests, and depending upon what the conclusions are, or the results are from those studies, we can draw better conclusions. But from this study, at least we can draw these conclusions. Smoking increases both levels of damage markers. 5-hydroxymethyluridine is the best indicator of current damage. 8-OHDG levels are increased in ex-smokers. Different types of DNA damage are repaired at different rates, with 5-hydroxymethyluridine repaired much more rapidly than 8-OHDG. Those we can draw from our little study. The reason I went through that much detail in that particular case is it gives you an indication of the kind of stuff that really needs to be done in order to validate some of these biomarkers. It's not enough to just think that a biomarker is good. If an oxidative stress marker is up, that must mean it's bad. It's a much more difficult, much more complicated, much more subtle process. And we have data, for example, that show giving antioxidants will drive down some markers of oxidative stress, but actually drive up other markers of oxidative stress. So you have to be careful about what you're doing when you're doing it in the dark. And that's one of the reasons we use the biomarkers so extensively, to follow exactly what's going on in our patients. Maintaining youthful hormonal balance, I won't stress this too much. This is kind of a ridiculous slide, but I'll point it out anyway. We show, in the case of hormones, an age distribution of the hormones. In this case, none of these hormones show any change as a function of age, so it's kind of useless. But in the case of DHEA, for example, where there's a difference as a function of age, you're able, by looking at this little postage stamp on the report, to see not only how you compare with others your own age, but with people of all ages, and that's kind of useful. Our approach is pretty straightforward with hormone replacement. We use estrogen here as an example. When we identify levels that are low, we restore them to youthful norms using bioidentical natural hormone replacement. I would like to comment briefly about the Women's Health Initiative. That's the recent study that was terminated because they got results showing that estrogen replacement was actually doing harm to the people receiving it. It's a little bit puzzling why they stopped that study, because none of the effects that they observed were not predictable by studies that had already been done prior. Everything that happened in that particular study had been observed previously in other studies, and yet they terminated the study prematurely, causing a lot of concern and consternation among patients and physicians out there in the world taking estrogen replacement. The reason I want to mention it is because a lot of people in the anti-aging community have jumped on that and condemned the study based on the fact that it used Prempro, which is synthetic estrogen. And although that might be the explanation for some of the effects, it's certainly not the explanation for all of the effects. And I think it's dangerous for us to take a position on something as complicated as estrogen replacement, especially given the data as it exists out there right now, and pretend that we know exactly why they got the result that they got and why it's still okay to take estrogen replacement as long as you do bioidentical and transdermal and whatever that is, because the reality is we really don't know. And it's going to take a few more years to really tease out the details. What I will tell you is this, that one study does not make a scientific fact. You have to look at the preponderance of evidence, and there is lots of evidence on estrogen replacement. And the bulk of that evidence suggests that estrogen replacement, even using Prempro and synthetic estrogens, is beneficial overall. And in fact, my best measure when I'm looking at any technology is what is its impact on all-cause mortality. And I used to say, I don't say it as strongly anymore because these last few studies on estrogen have kind of dampened my enthusiasm, but I'm still enthusiastic about estrogen replacement because it is truly, based on the evidence that exists today, an anti-aging hormone. It reduces the risk of death from all causes in patients who receive it, in spite of the fact that it increases risk from cancer, various types of cancer. It's too complex an issue to go through, but I just wanted to mention that. Not to slight the men, I always show this slide as well. We do look at testosterone, we do identify people who are low, and I think it makes sense to replace them back to youthful norms as well, with the caveat that if you notice, the y-axis here is the free testosterone index. We have historically used the free index because the free testosterone assays that have existed out there in clinical reference laboratories, including ours, have been lousy. They don't work. They don't give you any clear indication of bioavailable testosterone until recently. And there is an assay that's available now. We're currently setting it up in our laboratory, which is a radioimmunoassay for free testosterone, and that procedure will give you a clear picture of bioavailable testosterone, so that when you monitor that, you will be getting a reading on what physiological impact that testosterone level is having on your patient. So not all free testosterone tests are created equal, and at last year's Endocrine Society meetings there was a huge discussion on that very issue and a resounding condemnation of the clinical reference laboratory industry for its promotion of essentially bogus free testosterone tests. Nutrition. We do care about nutrition. Any anti-aging program has got to address nutrition somewhat. I was opposed initially to getting involved in the nutrition game simply because I didn't know much about it, and it was very confusing to me. And as the chief scientist at Kronos, the chief operating officer basically said, "Tough, Chris, go figure it out and tell us how to do it." And I spent months at the library at UCLA reading the nutrition literature, trying to make sense of it, and it was very difficult. You can find studies that will support any position you want to take about any diet nutritionally that you want. High protein, lots of data, lots of studies. Low carbohydrates, lots of data, lots of studies. High carbohydrates, lots of data, lots of studies. Low fat, yes, it's just a mess. In sifting my way through all these data, I finally came to the realization that the one common denominator in every case where they actually were seeing beneficial effects was reduced caloric intake. It's back to the old stuff that we learned in biochemistry 30 years ago. Calories do count. Reduce caloric intake. In other words, you can sum up the entire nutrition program in 2 words: eat less, especially in the United States. There are no magic diets. People attribute the longevity of the Okinawans to this amazing diet that they have over there, and how they eat is really the secret to their longevity. And it's a natural thing to look around at populations, find populations that do tend to live longer than the rest of us, and focus on what they do in terms of their diet to see if that's the reason. But the reality is that's probably not the case, because if that were true, then how do you explain the fact that when you look at populations or subgroups of people around the world, you find that the longest lived people on the planet are Chinese Americans. Now these are Chinese, and they eat American food. They just don't eat quite as much of it as the rest of us, but they are the healthiest, longest lived people on the planet. So making too big a deal about the ratios of macronutrients in a diet, I think, is not very constructive. I think what's important is to focus on making sure that in your diet you eat a sufficient variety of foods with sufficient attention to the macronutrients, that you get enough protein and enough of the omega-3 fatty acids that are essential fats, and focus on getting lots of fruits and vegetables and the nutrients that we know are important for health and longevity, and try not to get fat. The one thing you can do in terms of a biomarker to monitor how a person's diet is to monitor the red blood cell fatty acid profile, which we do. And that gives you an indication of the ratio of the various types of fatty acids, including trans fatty acids and saturated fats and monounsaturated fats, and the various types of polyunsaturated fats. And there's some value to doing that, but beyond that, it's very difficult from biomarker tests, even though there are some sophisticated tests out there that look at amino acid mixes and so forth. They're all pretty meaningless in terms of giving you an indication of a person's dietary habits over a long period of time. They can tell you a little bit about what they ate the last meal, but not much beyond that. So they're not very useful for that purpose. Last but not least, we look at exercise. And exercise, again, was something I wasn't very excited about getting into from an anti-aging standpoint, simply because I think there are a lot of people out there in the exercise business that do a pretty good job. But we were kind of forced to get into this by our patients who want to know how to get the optimum value from exercise with the minimum amount of effort. And so we've attempted to do that. And we do that by measuring various parameters of fitness that we think are important. Actually, this definition of fitness, including cardiorespiratory capacity, body composition, muscular strength, muscular endurance, and flexibility, is a classic definition of fitness that comes out of the exercise physiology literature. The reason it's important is that It's probably the best predictor of remaining lifespan of anything that you can measure from a biomarker standpoint. There's really nothing that does a better job than measuring in a quantitative fashion your fitness level, and particularly the cardiorespiratory function. We do treadmill testing. The neat thing about stress testing is that if you take two individuals and you set them side by side, and you can test them any way you want, and you get numbers back that give you some sense of what their health status is, but unless there's some serious problem that's identifiable based on those numbers, you can't tell too much about how fit they are and what their remaining life expectancy is likely to be. You take those two people and you push them to the wall, now you're getting at their reserve capacity. It gives you an indication of just how much reserves they have. And that's really what happens as we age, is that our resting capacity is pretty constant, but our reserves begin to decline. And we only die when our reserves fall through that threshold and drop below what is required to sustain life. So when you push somebody to measure their reserve capacity, it gives you a really good indication of what their health status is. So I really like, I had to fight very hard to get treadmill testing because everyone's afraid of it, and we don't do submaximal testing, we push them to VO2 max, and people don't like to do that. But the information that is obtained when you do that is so good that we do it. We do also measure body composition using DEXA. In your clinics, if you don't have a DEXA machine, there are a number of good ways of measuring body composition that aren't quite as good as DEXA, but the important thing is not so much that you measure it and get precisely their body composition. The important thing is that you get a number which is reproducible so that as you follow your patients over time, you can measure their progress. And that can be very valuable as a reinforcement for their staying on the program and continuing to show benefits. Measuring muscular strength is also quite easy. We use a hand grip strength dynamometer, and you can, with a number of different apparatus that are available, measure various other body parts fairly simply and straightforward. Hand grip strength at age 55 is the single strongest predictor of independent living 25 years later. So that's a very good indication or indicator for how a person is doing, and it's very simple and easy to do. Muscular endurance, well, muscular strength is your 1 rep maximum. So you find out what the maximum weight you can lift going through a full range of motion of a specific exercise. To measure muscular endurance in that same muscle group, you go back to 3/4 of that weight and have them do as many repetitions as they can until they can no longer maintain form, and that's their muscular endurance. And that's also it's one of the components of fitness. And last but certainly not least is flexibility. And again, there are a lot of fancy machines out there that you can buy that will enable you to quantify flexibility quite well. But there are a lot of very simple procedures that just involve sit and reach and things like that that work quite well for gauging flexibility. The bottom line is that if you put together a program for your patients that helps determine or detect disease early as possible, reduces their level of oxidative stress to the extent that you can, that helps them maintain youthful hormonal balance, that gets them to eat a little bit less, and the less the better really, assuming they can maintain proper nutrition, and gets them off the couch and around the block, exercising a little bit every day, you'll go a long way to helping your patients live longer, healthier lives. And that's what it's all about in terms of the application of anti-aging medicine today. Thank you. Do I have time for questions?